Concept 17.5, Mutations. In this concept, we will be going over what a mutation is, the types of small-scale mutations that we know about, and mutagens. A mutation is any change in the DNA code. It is not a change in the RNA, which is a temporary copy, or in the protein. A protein can be mutated, but it can't be a mutation. Here is a human example. All humans have a gene for hemoglobin, a protein in red blood cells that carries oxygen. The bulk of humans have what is called the wild type version of this gene, where CTT is the codon at this particular spot. In some alleles, a mutation or change has occurred in the first T that changes the T to an A. This results in a change from glutamine to valine. This results in hemoglobin that crystallizes when exposed to a lot of oxygen. This type of mutation, where one nucleotide is exchanged for another, is called a base pair substitution. Let's look at the different types of base pair substitutions that occur. So here is an example of a wild type code of DNA and the protein that results. So here we have an A instead of a G in this position. In the wild type gene, a glycine occurs in the protein here. When this mutation occurs, we still get a glycine. We call this a silent mutation. There is a change in the DNA, so there is a mutation, but there is no change in the amino acid sequence. So the protein remains the same and is not changed. This is a silent mutation. What if we have a T instead of a C at this position? Instead of a glycine, like in the wild type or in the silent mutation, we get a different amino acid, a serine. This is like the mutation that results in sickle cell hemoglobin. We call this a missense mutation. Think of it as a misspelling. You have a change in the DNA that results in a change in the amino acid sequence. Now this can result in a better, faster, stronger protein, but oftentimes it doesn't. Most of the time it results in a protein that doesn't function as well. So most missense mutations are bad. So what if we get an A instead of a T at this point? Instead of getting a lysine next in the amino acid, we get a premature stop. Not all codons code for amino acids. Some of them are like punctuation marks. A stop codon is like a period that says, stop the sentence here. So we have a premature stop and a prematurely shortened amino acid or peptide. In this case, we have just one amino acid. This is called a nonsense mutation. It's kind of like the sentence not making any sense, and the protein is non-functional. I generally refer to this as a killed gene. We still have the DNA portion, but it doesn't code for a functional protein. So these are all nucleotide pair substitutions where we've changed just one nucleotide. Let's talk about why we get silent mutations sometimes and why we get missense mutations. If you'll notice, this base pair substitution occurred at the end of the codon. We have an awful lot of codons that code for the same amino acid, and they all vary at this third nucleotide. This has led to the wobble theory for tRNAs. In other words, that the tRNA binds more strongly with the first two nucleotides between the anticodon and the codon, and it kind of wobbles on the third one. This allows for mutations to happen that result in silent mutations. This one, instead of occurring at the end, occurred at the beginning and resulted in a change or a missense mutation. This one also occurred at the beginning and it resulted in a stop mutation. So generally speaking, if a mutation happens in the first or second codon, it results in a missense or a nonsense mutation. If it occurs in the third nucleotide, oftentimes it results in a silent mutation because of the wobble effect of the tRNAs. Now let's talk about a different type of mutation. This is where instead of substituting a different nucleotide, we add another nucleotide. This one would be an addition, this one would be a deletion where a nucleotide is missed and the DNA code is shortened. Both of these are called frame shift mutations. Okay? What happens is all of the codons further down are changed because of that addition. I call these scantron mutations. It's kind of like if you're taking a test, you're doing great, you know all the answers, but you accidentally skip a bubble. Even though you knew the answers, all of the rest of the answers are wrong because they're one off. Very frustrating in humans taking tests. It's oftentimes lethal in organisms. Now, if you subtract an entire codon or triplet, like 3 nucleotides, you can remove an amino acid, but you don't change everything downstream. No frame shift mutation, okay, but this is a deletion. Same thing can happen if you add 3. This is called an addition mutation, but it doesn't result in a frame shift mutation. See how that works? As long as you put an entire word in, so to speak, or take an entire word out, you don't change the words or the codons further downstream. Mutations occur on a regular but rare basis from mistakes made during replication. There are some agents called mutagens that cause an increased frequency of mutations. Radiation is one of the more famous mutagens. You've all heard about UV radiation causing skin cancer and X-rays causing other types of cancer. So, how does it do it? Let's look at UV radiation. So here's a normal DNA and you have 2 A's and 2 T's next to each other. UV light has energy. It's just like the light that has energy in photosynthesis. And just like in photosynthesis, when the UV light or radiation comes in, it adds energy to molecules. In this case, it's added energy to these 2 T's next to each other. Instead of holding hands or hydrogen bonding with the adenines on the other side, these thymines now are bonded to each other. We call this a thymine dimer. This causes a bump in the DNA. So when the DNA polymerase comes along to replicate this, it can't get its active site over the bump. So it either kind of stutters here and causes an addition, or it just skips over the whole thing and causes a deletion mutation. In humans, this can lead to cancer. In unicellular organisms, this can lead to death. Chemicals can be mutagens. You're familiar with chemicals like those in tobacco smoke that are mutagens or can cause cancer in humans. Let's talk about base analogs, which we use in some beneficial drugs. See, here is a diagram of acyclovir. It has a nitrogenous base, just like the nitrogenous base in guanosine, but it doesn't have the ribose, it doesn't have the sugar. DNA polymerase thinks it's a G because of the nitrogenous base and tries to put it in, but the DNA polymerase can't hook anything to it because it doesn't have a sugar. This shuts down DNA synthesis, or it causes a deletion, both of which can cause death in microbes like herpes viruses, or it can cause death in cancer cells that are dividing uncontrollably and are continually going through DNA synthesis. Some chemicals change the bases. Nitrous acid can change an A to a hypoxanthine, which looks like a G. This causes a base substitution mutation. We don't use nitrous acid in any clinical situation, but We are aware that it can cause mutations, and we try to avoid it in our daily lives. Well, that's it for this concept. Once again, make sure to write down your questions so that you remember what your questions are when we have question and answer in lecture.